Yeah, that's good.
Yeah. Okay, perfect, perfect.
Yeah.
Okay, so let's get started. So I'm gonna tell you a little bit about quantum dynamics and biology. And as I promised also, there will be, although really I'm a theoretical physicist, so I do everything with mathematics in the end. In this lecture, I think I've made sure that there's a grand total of 0 formulas. So I will be, of course, a little bit more general in some places, but everything that I'm telling you about underneath lies a whole bunch of mathematical formulas and rigorous methods that lift this from the general, let's say, conceptual point also to really rigorous mathematical proofs and numerical methods and so on. Right. So, and before I start, yesterday I had a brief trial session with Nina, and we came to talk, and I wanted to, and she encouraged me also to spend a few words about this building that you see in the background here. And that's the Center of Quantum Biosciences here at Ulm. And this is something that I started to put together in 2011. So, and the central goal here was to bring together strengths of this university. And those are, on the one hand, quantum physics, and on the other hand, medicine. So there's a very good medical school here in Ulm. And I already, when I was at Imperial, I had developed this interest of trying to work at the interface between quantum mechanics or quantum physics and biology. And there it proved difficult to bring people together. But here, this proved to be more successful in the end. So in 2011, I went to the president of this university and I said, basically, this is what I want to do. I want to bring together physicists, chemists, biologists, and medicine people in some facility which has really tailor-made labs, also state-of-the-art labs, where you can do quantum physics and biology experiments in one lab and bring methods together and so on. And then I started to, they supported this, and I started to find the money. And fortunately, the federal government gave a lot of money, 27 million euros to build this building. And so it's completed now. Since 2019, we started to move in really fully in January, so then the activities got a bit interrupted. But we have here really theoretical physics, experimental physics, and I will tell you also a little bit about why we have these experimental physicists. We have chemistry and molecular medicine groups here in one building, very close together. And indeed, we have laboratories where a quantum physicist can put his lasers and whatever methods they use, and medical molecular medicine labs, and where they can really work together, make experiments where we use experimental physics methods on biological systems directly to try and bring out really quantum mechanical behavior in these biological systems and prove that these behavior exists, right? And so this is, so we're at the start now, of course. Now we have to show what we can do, but it's really a nice, it's a nice setting now to really work together across disciplinary boundaries, right? And so this is, this is where I'm sitting right now also. Right now, right below me, one floor down is the molecular medicine and the chemistry. Right next to me is an office full of experimentalists. So, and we meet each other all the time, and we have joint projects and work together. So that's a little bit about this Center for Quantum Biosciences here. But now, what are we doing here? What are some of the things that we are doing? And one of the things that we are doing is really trying to explore this interface between quantum physics, quantum dynamics, and biology. And this has 3 aspects to it. And one is we really want to find out what role does quantum mechanical behavior play for processes of life. So, and that's one of the things that we want to find out here, both theoretically and experimentally. And to really explore this question, not only theoretically, but also really prove things, that means doing experiments, we really need to develop new measurement technologies as well. And here, what we are also doing is we are using particular type of quantum technologies that will enable us to look at individual biological receptors, for example, and examine their properties with atomic scale resolution to try and see whether there are really, whether there are single electron transfers, for example, and whether these are really quantum mechanical in nature or merely classical processes. And so I will tell you at the end of the lecture a little bit what this technology is and what we have in mind here. And then also maybe one of the upshots of this whole thing will be that, of course, we understand more deeply life or processes of life, but maybe we can also turn these things around then and use biological systems and quantum phenomena in those to make them better sensors that we can use maybe in technological applications, but also maybe to understand how nature actually, how biological systems, sense external stimuli in new ways. So these are the kind of the three goals that we have for our research at the interface of quantum dynamics and biology. And maybe the fundamental starting point, so why I certainly got interested in that, is maybe encapsulated in this graph here. So here on the right upper corner, we have, well, let's say the brain, so very macroscopic, very large object. And really, I mean, I certainly believe that all the essentials of the brain function will be understandable using classical physics. So I do not really believe that that we will need quantum mechanics to understand consciousness, for example. Yeah? So I think this is probably a classical phenomenon. But then we know that if we use physical tools and we examine structures in finer and finer detail, we see that the brain, of course, is composed of sort of neurons and synapses, and if you go further down, and then in the cell membrane you have receptors, and you go further and further down. And at some point, we know that when we reach the level of individual atoms, then really the laws of nature change, or let's say other laws of nature become relevant, and namely those are the laws of quantum physics. And in these realms, at these very, very short time scales and length scales, we don't have particles or waves, but actually these objects can behave both as particles and waves at the same time. And this leads to new phenomena. So for example, an electron will not only be a point particle, which we can locate in one place, it will also have a wave characteristic whereby it's spread over an entire spatial region in a coherent fashion, almost like a water wave. And these new laws of nature were discovered in the 1920s, roughly, so 100 years ago, pretty precisely. And it led us to a fundamentally new understanding of how atoms and molecules behave, and they really underpin all of our understanding of chemistry in a way, the fundamental understanding of chemistry. And so we know that up here, the world is really well described by classical physics. Down here, we need to use these new laws of quantum physics. And so somewhere in between, there must be a transition region, right? And so somewhere there has to be an interface where the world is both quantum mechanical and classical at the same time. And if we are at this interface, it might be easy to move here and make everything classical, and everything behaves like point particles, like little footballs or soccer balls that move around. Or we go here in this region, and then really the wave character of things also become important. And we really don't know precisely where this boundary actually is. And so one of the questions that motivated me to go into this was to see, is this boundary here that were indicated in the range of really macromolecules, very large molecules? Or is it actually really down here where it's only relevant for, let's say, very small molecules, where it really only describes chemical properties or so of systems? Right, so one question was, where is this boundary? And we don't know really. So we have to actually now find out by experiment and theory where this boundary really is. But then there's another question now. If we are at this boundary here, then actually it might be easy and of interest for nature to actually shift it, to try and, for example, make its properties a little bit more conducive to support quantum mechanical properties, or to make the system more noisy and more classical. Yeah? So it might be that in some processes, nature will, or biology will adapt its structures in a way that the system might be a little bit more classical, really. And maybe for other processes, it might be more advantageous to use a bit more quantum mechanical features, and therefore, for example, isolate the system a little bit more from environmental noise, and therefore make it more quantum mechanical. And so it might well be that if we are sitting at this boundary, that this is a place where nature will have an additional control mechanism whereby it can use either these laws of nature or these laws of nature to its advantage, right? And so we want to find out where's this boundary, and we want to find out whether indeed nature is trying to adapt its processes to shift this boundary a little bit, depending on what it wants to achieve. Okay, so these are kind of fundamental questions that we would like to understand better. And so that's fine. And so then, of course, as I said before, up here we believe that many things, most structures are explained very well by classical physics. But of course, if we have quantum mechanical laws making certain processes, biological processes, more or less efficient, it will nevertheless have an indirect impact on these larger scales, right? And so this is another thing that we would like to understand. If we can make processes more or less efficient here, what relevance does it have to processes at larger scales? Okay, so these are general goals. So, I mean, the way I like to look at biological systems is, of course, from the viewpoint of a physicist, really. And so for me, biological proteins and so on are really small machines. This here on the left-hand side, we have a large machine. That's a classical engine with all sorts of components that we can identify very well and which have a pretty large volume compared to their surface. And so there's a little bit of friction involved. But largely, I mean, certainly we can take this machine, we can look at its components, and we understand it very well using just classical mechanics properties. And we want to take this picture and we want to look at proteins also from a point of view of them as thermodynamic engines that take an input, produce an output, have some interactions with an environment, but now they might do this using some quantum mechanical processes as well. And so this is what changes here is, of course, that these processes will be faster. Our normal sort of mechanical engine will run at at 1 turn per second, like a clock or so, something like that. And so the processes that we will be looking at in the biological setting will be much faster. So the slowest process that I will possibly consider will be a microsecond. But more typically will be nanosecond, picosecond, or femtoseconds. So very fast processes. Yeah. The systems will be much smaller, and therefore also distances that need to be covered will be shorter. And so I really would like to generally talk about processes that are on the, let's say, nanometer scale, 10 nanometers, tens of nanometers, but not really very, very much larger than that. Because then actually it becomes difficult to cover these distances at these very short timescales that I'm interested in, right? So this sort of sets the length and timescales of processes that I'm interested in. And the other effect that will happen here is that when I make the system smaller, then actually the surface to volume ratio changes. So actually the surface becomes much larger relative to the volume than as compared to these classical machines and on the left upper corner here, right? And that means that fluctuations grow. because you're interacting more strongly with your environment. You can randomly interact with components that come in touch. There can be collisions. There will be more noise in the system. So on the one hand, by making systems smaller and faster and so on, they allow for more potentially quantum mechanical behavior. On the other hand, the noise is also growing, and this tries to suppress quantum mechanical dynamics. And so there will be an interplay between these two things. And it's quite clear from all the research that we are doing that there can be a very fruitful interplay between, on the one hand, having this enhanced fluctuations and noise and these interactions with the environment, and on the other hand, this quantum mechanical wave-like behavior. And really what comes out of our work is that the best place to be for many processes is when you have both, when you have a certain amount of noise and a certain amount of quantum mechanical wave-like behavior. And that is really exactly being at this boundary between the classical world and the quantum world, right? And so that's what I want to aim at in this lecture. I want to explain you a little bit more which processes might be interesting for us, where we have a very good reason to believe that quantum mechanics does play a role for the functionality of biological processes. And in each of these processes, I actually want to highlight the fact that they are really at their most efficient when the system is not like a pure quantum mechanical system, like an individual atom or so, but really when there's also some environmental noise that plays a role as well. So these two things have to come together, and maybe that's not a surprising thing, because in our body it's, you know, hot, warm, humid. I mean, there's lots of collisions and there's all sorts of stuff around that interacts with each other. So nature really has to deal with these fluctuations and this noise. But on the other hand, at the atomic scale, there's quantum mechanics, which can in some cases give us advantages, and nature will probably want to make use of those advantages. And so there is automatically this collision of these two effects, and it's likely that nature has made optimal or near optimal use of these phenomena. Okay, so what are the building blocks that I will typically be considering? And so on the one hand, we will have protein structures. So that's on the left upper corner here. And so they provide our scaffold to put things together. And on the other hand, in many of these examples here, I will have molecules that will support either electronic excitations or spin degrees of freedom, so little magnetic moments, right? And now what these proteins and these molecules, when we put them together in controlled arrangements, what we are really doing is we are trying to build little electronic networks. Okay, so we are putting places where, for example, electrons, or electronic excitations or spins can sit, and they can hop from one place to the other. And the way in which they can hop from one place to the other will depend on the spatial arrangement of these molecules that we place in this protein scaffold. Okay, so in that sense, nature makes these controlled arrangements and builds little electronic networks. Okay, and of course, what we will have here is not everything is static here. In quantum technologies, we would normally aim to have everything very, very static, very stiff, not moving, no environment. This is not the case here in biological systems. So we have this protein, we put the molecules in there, the scaffold, but everything will move a little bit. And that means that there will be a degree of uncertainty, a degree of variation, and a degree of fluctuations. And it's really thermal motion to a large extent that brings in these fluctuations into the system, right? And this is unavoidable. Yeah. Okay, so what will I be looking at? And I will give you some example from the world of photosynthesis. Not necessarily because I believe that this is the most exciting area, but it has the advantage that it's the area where we have the most direct experimental proof that actually quantum mechanics does play a role for these, for fundamental biological processes. And this is because these are very fast processes, but they can be examined by light. And we can send very, very short laser pulses onto the system and study the reaction of this system. And we can do this so fast that we can really see the effect of quantum mechanical dynamics in those systems. And we can show that it does make a difference to have quantum mechanics and noise and both of them together. And so this is, in a way, it's very encouraging because we can see there that nature does use these processes. Also, we have very strong evidence for that. But then I would like to go actually further and take some lessons that we learn from these ideas and go to an idea that is much less well examined experimentally. It's much harder to do this because it's really a purely electronic process. It's much more difficult to see electrons in these systems. And this is the idea that perhaps electron vibrational coupling can play a role in our sense of olfaction. Okay, and I will explain you why this might not be such a crazy idea. We don't know whether it's true or not, but it might be, it's certainly an interesting idea. And once you sort of buy the idea, similar ideas in photosynthesis, you would say, okay, why not in olfaction? And then we have to think, of course, how to do this experimentally. And this will be at the end of the talk, some ideas of how one could go about this. And then I would also like to touch upon another point, and it is magnetoreception. And this is now, it's also an electronic phenomenon, but it is not about the charges of the electron, but it's actually about the magnetic moment of the electron. And again, this is a very well founded theoretical idea. So I mean, for a theoretical physicist, when it's described to a theoretical physicist, it would say, yeah, why not? It can very well happen. But the challenge, of course, will be to find out, both theoretically and experimentally, whether the conditions in biological systems are really right for these processes to happen the way we theorists think they might be playing out here. And these are the three examples that I want to show you. And it would then be interesting, actually, also for me to see where processes of this type may also play a role. For example, in mitochondria or in other electron transport processes or so, where might be good regions to look whether some of these ideas may also play a role. Okay, so and that would be great also for the discussion afterwards to see, you know, where we can move to from in our interest as theoretical physicists. Okay, and so. In all these three processes that I will talk about, there is the following general observation. And you see in the background, I mean, I said 0 formulas, but I mean, okay, so here's a little bit of a formula. This is just meant to show, okay, there is a mathematically rigorous foundation for all these things. But what we find both from analytical theory and from numerical examinations is the following general observation. Here is performance of some process in biology. And I deliberately don't put units. It doesn't really matter here. This is in some arbitrary units. And here I put what I call system environment interaction. So by that, I mean, I have on the one hand, I have, let's say, my electronic degrees of freedom. These might be charges, or might be my little magnetic moments of the electronic degrees of freedom. And they will interact with each other, right? Often in a very coherent quantum mechanical fashion. But they will also interact through the surfaces, let's say, to other uncontrolled degrees of freedom, like water molecules, noise that there might be. And so this interaction, system environment interaction, is really the noise that is coming in through finite temperature and all these sort of things that the biological system has naturally. And what we find typically is that when we have very, very strong system environment interaction, so high temperatures, a lot of solvent, a lot of liquids around, a lot of motion or so, then the process that we are looking at is not very efficient. If on the other hand, we say we have really none of this noise, we have a very purely low temperature quantum mechanical, purely quantum mechanical system, well isolated from all the environment, then actually it may also often not do the job very well. Okay, it becomes less efficient. And we find typically that it becomes particularly efficient when we are sort of in a halfway house between where we have interactions between our system components, which are roughly of the same strength as the interaction of each of the system components with the environment. Yeah, so that's a qualitative statement. I mean, this can sometimes be better that it's the system components interact 3 times as strongly as with the environment, or maybe only half as strong. But let's say ballpark figure is that both of these interactions are of the same level. Yeah. And so that's an observation that we find, and this actually holds true in photosynthesis. It holds true in this purported mechanism for olfaction, this quantum mechanism, and also it holds true for this magnetoreception. So it seems that all of these phenomena where we use quantum mechanics, it is always helpful to also have really noise around, which is good because you cannot avoid noise in a living biological system. Okay, and so now what I want to do is I actually want to show you first at the example of photosynthesis that this is actually, at least for photosynthesis, this is true. This is not merely a theorist statement, but actually this is something that we see in experiment. Okay, and that's the first point that I want to bring home. And then once I've done this, I want to show you how we can turn this on its head and turn this into a biological sensing device. Okay, so and now you see really that I'm a theoretical physicist because I abstract things quite a bit and make things more simple. And in the proper analysis of things, then of course we put all the biological complexity in again. So we try and take into account as much as possible the true experimental parameters that we have from experiment. The true fact that we have a protein surrounding all these things, that it has thermal motion and all this sort of stuff. But for the principle, I would like to make things really very simple, oversimplified actually, but it brings out the basic idea. And what is a photosynthetic complex in its most simple setting? Well, we will have a place, the reaction center, where actually in the end, electronic excitations arrive and where they will be ripped apart, so there'll be separate charges, and this charge separation will be stabilized, and it provides then in further steps the fuel to drive subsequently some proton transport and all sorts of other processes. But for us, important is that this is a place where we want to be, where later on chemical energy in the end is produced. But the energy comes from the sun, so it is actually absorbed in quite a different place. So the reaction center is very small. It is only a few nanometers maybe in size. And this is the reason for this is that we really have, the dynamics there depends on electron tunneling, which is only effective over very short distances. And so this is a very compact unit. It's very small, and therefore itself, it can absorb very little light because it's so small. Cross-section is very small. And so you need to build around this something that is like an antenna that helps you absorb light because it's large. And therefore, it has a very high chance that somewhere in this large object, a photon from the sun will be absorbed and creates an electronic excitation. Yeah. And then we have to have something in between, namely we have to have this transport process which takes this electronic excitation and moves it into the reaction center, where we need this excitation. So we have a transport process in between. Okay, so what I will be talking about now will be initially the transport process, but then I will show you later on, there'll be very good experimental evidence that also this charge separation here very much benefits from both the presence of quantum mechanical behavior and noise. Okay, and really experimental evidence actually. Okay, so but what do we want to do? So in the first step, I want to make sure that this transport process is very efficient. Yeah? We want it to happen very fast, very rapidly, so that it's sure that this electronic excitation doesn't just dissipate to the environment, but actually really brings its energy to a very specific point, namely the reaction center. And so we shouldn't be too slow. Okay. And so for that, nature has made these pigment protein complexes. So it has this protein, and in it, it holds these small molecules, pigments, that can support this electronic excitation. So the electronic excitation will come and propagate through this system along various paths into the reaction center. And now we wish to tune this network in such a way that it does this job as quickly and as efficiently as possible. Okay, and what degrees of freedom do we have? What can be changed here? We can change the relative distances between those molecules, and we can change also the relative orientations. So the relative distances will actually affect the rate at which an excitation can hop from one place to the other, and it decays roughly with 1 over the distance cubed. So it carries quite a significant penalty if you move things apart too far. Yeah. The orientation will also affect the rate at which excitations can jump from one place to the other. So if the molecules are nicely parallel, typically this jump rate is, or this transfer rate is larger. If they become orthogonal, this rate becomes smaller. Okay, so there's really a relative orientation phenomenon here taking place. Okay, and so these are the degrees of freedom that you have. And of course, in addition, everything is shaking. So that means also the energy it takes you to bring an excitation from one place to the other might differ in time and might differ also in space along this network. Okay, and so these are the things that these are kind of the things that we can change. And now the question is, how do we make the optimal, or how does nature make the optimal transport network? How does it look like? And for that purpose, we have to also take into account one other degree of freedom, and that is actually the presence of molecular vibrations and protein vibrations, which form our environment, right? And so there's this, all the time, we have this shaking and motion of this entire complex at a whole range of frequencies. And they will actually extend to, let's say, okay, a few hundred wave numbers to at most a few thousand wave numbers. So what does that mean? So these are frequencies that are much slower than, let's say, the oscillation frequencies of light. Typical ranges of these frequencies are, let's say, periods are maybe 10 femtoseconds up to maybe a few hundred femtoseconds, right? So this is the vibrational frequencies that you have in the environment. So this is the rate at which all these structures will fluctuate. Yeah? And so that means that whenever we have here energy levels in the system, energy differences that are of the same order. Yeah, so let's say there are frequency differences between different sites here that are of the same order as frequencies that are happening in the environment. Then we have a resonance, and energy can also be exchanged between the system and the environment. So that means, oops, that's too early. So it means that actually we can go from up here, from this higher energy state to a lower energy state and get rid of the energy difference into the environment. Yeah. Or alternatively, if we are sitting here and we have some energy in the environment that oscillates exactly at this energy difference here, it can actually give me some energy and move me up here. So I can go from down here to up here. So these are processes that can also take place. And so now what we can do is we can try actually and adjust all these energies, electronic energies in the system, by moving the molecules closer and farther apart from each other and changing their relative orientations. Every time I do this, I will change this energy level structure. And to give you a simple idea of why this happens, I mean, a classical physics idea, this is where this picture comes in. So now let's talk about some simple pendula. Really think about, like if you have a nice pendulum clock in your office or so. I mean, it has it oscillates back and forth. For example, here in Jeffrey's office, and so let's say we have two of these pendula, and if they're completely independent of each other and they're two good clocks, they're made by a good clockmaker, they will swing pretty much at the same frequency for all the time. Yeah. And so whether they are out of phase or in phase will make no difference to their frequencies. So if they are moving in phase, they have a frequency of 1 tick per second, let's say. And if they are just out of phase, they also go back and forth in 1 second. So that's their frequency. But now, if I do a slight change here, and I attach to both of these pendula a spring to couple them, so that when they are trying to move apart, the spring is stretched, and the spring is trying to drive back the pendulum. So there's a restoring force. And now you can see that now if the two pendula are out of phase, they will be driven backwards. And so there will be a stronger restoring force. And so they will actually oscillate at a slightly higher frequency. Yeah, so that's an experiment that in every undergraduate physics lab, and even sometimes at school, one is doing. If there's some coupling between those, if they are moving out of phase, the frequency is higher. But if they're in phase, then the spring is never stretched. There's no extra restoring force. And so they are still going back and forth at exactly 1 tick per second, let's say, while the coupled springs will go back and forth maybe in 0.5 seconds. So they have a higher frequency. So introducing interactions and changing interaction strength will change the frequencies in your system. And that's exactly on the quantum mechanical level what we are doing here in this biological systems. We are changing interaction strength. Therefore, we are changing the frequencies of the electronic system. And therefore, we are changing the ways in which it can exchange energy with its environment. Okay, so that's an important basic idea. And I mean, of course, it's classical pendula here, but the principle, it's actually the same. And so if we have no environment, so we would create some energetic configuration. And now we start, let's say, up here with all the population in, let's say, in this side here, up here at high energy. Now you would say, okay, no problem. The electron catch or this excitation could just move downwards, try to go to lower energy. But actually now another physical principle comes into play, and that is of energy conservation. If I want to go from this high energy state to this low energy state, the energy difference has to go somewhere. The energy in the world is conserved. Yeah? If I lose it somewhere, something else has to gain. Otherwise, this process cannot happen. It's not possible for us to create energy out of nothing, and also not possible for us to destroy it. So therefore, really, to make these transitions here, I have to have some environmental degree of freedom that can take up exactly this energy difference. And so these are the vibrations in the environment. So we have this electronic degree of freedom that has energy difference and eigenfrequencies, which have a certain spectrum. And we have these vibrational degrees of freedom. And the vibrational degrees of freedom cannot be changed so easily. So this is given by the protein and the intramolecular vibrations. But what we can change is the energy differences in the electronic degrees of freedom. We can just change them. So we can adapt the electronic network by changing interactions to the environmental fluctuations that we have. Just like we tune a radio, basically, to try and be receptive to a particular frequency. And that's exactly what we're doing here. So if we have a lot of vibrations at a particular frequency range, we try and change our electronic network, our electronic frequency differences to match exactly these dominant frequencies in the environment. And if we do so, then actually we can take this state with a high energy, and it can go down, and in exchange excite vibrations in the environment. So the environment takes up some of the energy difference. And it only becomes possible if we can tune our network in just the right way. Okay, so this is a basic principle that we believe, and one can also see experimentally, is happening in photosynthetic complexes, whereby it tunes an electronic network, and this tuning is a quantum mechanical process, and tries to adapt it to a vibrational environment to make best use of this environment and the ability to dump energy in there, but maybe also to go, if you are trapped in this low energy state, to go up into that state. Yeah, and to take energy from the environment, bring it back into the electronic system. So, and when one goes through the analysis, then this really works best when we both have good electronic coherent quantum mechanical interactions, which allow us to tune the energy levels. But we also need some interaction with the environment. If the interaction with the environment, the vibrations, is very weak, then we cannot make use of it. We cannot efficiently and quickly dump energy into this. So we have to have a nice interface to this vibrational degrees of freedom to be able to get rid of energy differences. Okay, so we need sufficiently strong interaction to the environment. But if it's too strong, then actually it interferes very strongly with this tuning mechanism and with the interactions, and actually we lose the ability to tune our radio network. And actually this also happens in a radio. If there's too much sort of noise around, you will not actually be able to tune into any particular radio station. You will only hear sort of crackling noise. So there has to be the influence. I mean, you have to be able to see the sender and get the radiation coming into your antennas, but it should not be too strong. Okay, so now this is qualitative arguments. This can be proven mathematically. Okay, and then of course the question is, does this also happen in biological, really in photosynthetic complexes? Not only at a theoretical level, does it also happen in the experiment? Do we see this? And these experiments that we are looking at there actually take into account another phenomenon, namely that we do not only have a very broad range of frequencies, but actually, when you look at these small molecules here, I mean, small molecules are very stiff objects, so they vibrate at very specific frequencies. Yeah? And so they are very, very well-defined frequencies, and these oscillations also live a long time. They can live picoseconds, much longer than all the transport process that is happening. Yeah. And so what we wanted to see to prove that there is this beneficial interaction between the electronic degrees of freedom and sort of vibrations in the environment, we wanted to actually see the interaction between the electrons and this very specific long-lived vibrations, because then we would expect to see the following phenomenon, namely that we start at the high energy state, it goes down, it excites one of these long-lived vibrations in the molecule, but because the molecule, but this long-lived oscillation still holds this excitation, it also can give it back. So we should see actually an oscillatory behavior. The excitation goes down, gives it into the environment, then the environment gives the excitation back, and the electron moves up to its original state. And this should be a periodic process that happens several times. And if we see this experimentally, this oscillatory motion, then this would be a very strong evidence for this electron vibrational coupling taking place. That's the first step, basically. And that's exactly what we see in experiment, actually. And we see this in biological systems, but we also see this in actually organic photocells, which we also chose for some of the experiments simply because they are much cleaner and easier to examine. And there we also looked at the impact of all these interactions on the charge separation process. And so let's forget about the details here, and let me just show you the result. So here we had a process where we initially had a photon comes, it's absorbed, creates an electronic excitation, which moves to a particular place, in this case a boundary, and then is separated into positive and negative charges. And we wanted to see experimentally how quickly this happens in the presence or the absence of vibrational motion. And so then when we look at this, and you look here only at the black curve, you see in the real environment with lots of vibrations around and the specific long-lived vibrational mode, the charge separation process, this black line, is very, very rapid. In very short time, this is about 20 femtoseconds, the charges are separated. Now, what we can also do is we can remove this vibrational environment to some extent. And then we see again, only look at the black line, and now we see that this charge separation is much slower. It also happens, but it takes 10 times as long. So here, this is a clear experiment. Yeah? So this is a clear experimental proof that actually this interaction of the electronic degrees of freedom with their vibrational environment can really be beneficial for a very fundamental biological process, namely this charge separation. Okay, so this is here on an artificial system, of course, but similar experiments that are less clear cut can also be done in biological systems. And a lot of the current drive in experiments is to make more clean cut experimental proofs of this kind of phenomena in biological systems. Okay, so that shows you that the interaction of electronic degrees with vibrations has really a beneficial effect. And now I want to show you how this fundamental idea might actually be used as a sensing device. So far I had tried to make use of this environment to make the charge separation process or transport process a little bit more efficient. But now I want to go the other way, and in a way I want to use the fact that there is charge separation as or movement of charges as a signal for the presence of particular vibrational degrees of freedom, and thereby build a sensor. And that's the idea of using vibrational degrees of freedom in olfaction. And that's an idea that Luca Turin brought up in 1996, actually. It's basically the following idea. So here you have a cartoon of a, let's say, a membrane receptor. Yeah? So this would be the binding pocket of the receptor where eventually a molecule might end up sitting in for a while. And the rest here is the receptor. So again, I mean, I'm abstracting things for the simplicity of ideas. And in the end, one has to check very, very carefully whether all the conditions for these simple ideas are really present in biological systems. And so the idea is that in addition to this binding pocket, somewhere in this receptor, we have an electron donor and an electron acceptor. And they will have a certain energy difference. And so we start with an electron being ready here, up here, and it would love to move to the acceptor. But as I explained before, to do this step, to go from the donor to the acceptor, we have to be able to get rid of this energy difference to the environment. Okay? So there have to be some vibrational degrees of freedom that can take up exactly this energy difference. Now, let us assume that the receptor is configured in such a way that really this energy, there's really no vibrational degrees of freedom, or very few of those, that have this suitable energy, that can take up this energy difference. So if there's nothing else, if the receptor does not have in its binding pocket any kind of molecule or nothing else, the electron is trapped here and it cannot move. So now imagine a molecule is coming, sits in the binding pocket, and of course, this molecule will have vibrational degrees of freedom. And if one of those vibrations is very well matched to the difference, can take up the energy difference between this donor and this acceptor, then its presence will facilitate the electron transport. So no molecule, no transport. A molecule with the right vibrational frequency that has a quantum, that takes up quanta of energy, which are exactly the energy difference between donor and acceptor, will lead to this process. The molecule will get excited and the electron moves from donor to acceptor. And then of course, I mean, a charge transport in such a receptor may then change the electrostatic environment, and it will then lead to some further processes further down the line, and it can initiate some signal processing chain. But that would only be the first step, right? And so this is a very attractive idea. So you could actually have in this way a sensor that can discriminate between the presence and absence of molecules, and not only that, it can discriminate between the presence and absence of molecules with particular vibrational frequencies. And it would be very attractive if this would actually be a way by which a biological system would actually sense molecules. In fact, incidentally, this is a way in which solid-state physicists probe vibrational spectra of molecules. They try and drive electron currents through molecules and change the energy difference between donor and acceptor and look where is the transport good and where is it bad. Yeah. So this is actually a perfectly, as a principle, this is perfectly well possible. The question is are there do olfactory receptors, for example, have these donors and acceptors for electron transport? Are the environments sufficiently noise-free so that the electron doesn't move all the time, but only when a molecule comes into the binding pocket? And generally, is the coupling also between the molecules in the binding pocket and this electron transport system strong enough, right? And so there is a lot of things that have to come together, actually, to make this work in a biological system. But in principle, theoretically at least, if all the conditions are right, this can happen. So why is this? Might this be exciting? Why might this actually be relevant to olfaction? I mean, I would say the standard theory of olfaction is more one, we have different olfactory receptors. You try, I mean, they have different binding pockets and they might bind different olfactants. Different classes of receptors might be accepting olfactants in the binding pocket, and it's basically a shape-based mechanism, maybe a bit of chemistry involved. And so if a molecule binds to receptor class 3, 17, and 55, then this is relayed to our brain as this smells of musk or something like that. And so that's a perfectly fine idea, and it surely plays an important role. But there are interesting phenomena, namely, if you take molecules, olfactants, and they typically contain hydrogen atoms. And if you replace the hydrogen atoms by deuterium atoms, then actually these molecules can smell differently. Okay? And then the question is, why is that? Because chemically, this is not a huge change. It is a change, but not a very, very large one. But so you would expect that, well, why should there be such a difference in smell? But one thing that changes very, very significantly is the vibrational spectrum of this molecule, because you replaced hydrogen by something that is twice as heavy, namely deuterium. And that makes a big difference to vibrational frequencies. Actually, can change them by 40%. And that is a big change. And so it is an attractive idea, therefore, that perhaps there is a spectral component to our sense of smell, whereby it really is also sensitive to vibrational modes in the molecules. And then one could, if this would be like that, then one would of course need a mechanism for this to happen. And this is what I just explained, is a potential mechanism to make something like this happen. Okay. So here we have now turned basically these photosynthetic ideas a little bit on their head, and we have used the fact that electrons can move depending on the presence or absence of particular vibrational modes as a sensing device. So this, of course, can also happen in other electron transport processes. You can take any electron transport in your biological system. You can now ask, how is it affected by the presence or absence of well-tuned vibrational modes in the environment? Which conditions do you have to satisfy so that this electron transport is particularly efficient, or let's say is of such a nature that it realizes a particular biological function in the most efficient way? Sometimes this might be that you want to have the electron moving very fast. Sometimes it might be that it has to move very slow. And so you may also, by changing the vibrational environment, you might affect actually these electron transport properties. And on the other hand, you can also ask, not only can I change the vibrational environment, but can I also, through electronic interactions, in the presence of a fixed vibrational environment, can I change the electronic properties and the transport and the dynamics of the electronic system in such a way that it is optimal for our purposes, right? So these are questions that this poses. And of course, interesting would also be to see, okay, where in biology do we have maybe electron transport phenomena problems that are not really well understood, where perhaps such processes may play a role or may make a difference, okay? So that's one thing. And now I would like to touch briefly upon the magnetoreception part. I don't want to go into terribly much detail, because then I want to really go to the new measurement technology that we are trying to develop to actually shed new light on these processes for direct experimental evidence. So it's known actually that birds have a magnetic sense. So that's undisputable. They use, amongst other things, the fact that they can actually sense the inclination of the magnetic field against the Earth's magnetic field against the horizon, they use in helping them to navigate when they actually really take their many thousand-kilometer journeys from Northern Europe to Africa and back. They always find their way. And one of the things that helps them there is really a magnetic sense. That is well established. There's really no doubt about that. The European robin, I mean, many birds have, and many other animals actually have magnetic sense. What is much less well understood is how this magnetic sense actually comes about, how it actually is really realized in birds, for example. And there are various theories, and one theory is certainly that birds may have a small magnetite particle, so very small micron size or even smaller nanometer scale particles that are magnetic. And of course, magnetic particles act like a compass needle, so they will react to magnetic fields. And then if they are attached, for example, to an ion channel or so, it might stimulate the opening or closing of an ion channel. Something like that might be a mechanism. The problem is that there is really no particular evidence for this to happen in birds. There are these magnetite particles, but they don't seem to actually be in any way related to magnetic sense. So it's really a mystery what is precisely happening here. And so actually it was already in the 70s that Klaus Schulten, when thinking about actually chemical processes, realized that there might be a mechanism that is a chemical mechanism, but that may impart a magnetic sense to, for example, also birds. And so what was known at the time was that some chemical reactions will change their yield depending on the presence or absence of magnetic fields. Okay, so that's a fact, a well-established fact. And the question was, how might that happen? And so these were light-stimulated processes. And so the idea that Klaus Schulten had was that actually you have some molecules that would like to make a chemical transition. And the first thing that would happen is that light So you use light, you shine light onto the system, and it would excite electrons from its ground state to an excited state. So 2 electrons actually, and that's called a radical pair. And initially this radical pair, these electrons, and they carry some magnetic moments, but initially these magnetic moments would be exactly opposite, right? So in quantum physics, we would say they're in a singlet state, but let's say for our purposes, the magnetic moments are opposite each other. And so I have here a little picture, see on the left-hand side. So if you take 2 magnets and you hold them close together and with opposite polarity, then you will see they stick very well together. And if you hold them in your hand and there's let's say a compass needle nearby, it will not actually see very much of a magnetic field. No matter how you orient these 2 magnets, you will not see much of an effect because simply these 2 oppositely oriented magnetic moments compensate each other almost perfectly. That also means that these magnets will not really experience any force irrespective of how I orient them in the Earth's magnetic field. However, if I flip one of these so that 2 magnetic moments are parallel, then you will see that now these magnets have a very strong effect on your compass needle because they're not compensating each other anymore. And also if they are sitting in a magnetic field, and if it was a very strong magnetic field, then you would actually need a force to actually turn them, right? So if in this biological setting or in this molecular setting, there's a process that flips the magnetic moment of these spins from being anti-parallel, sometimes to being parallel, then the energy changes. And also this resulting magnetic moments are very sensitive to the external magnetic field. And also the energy will depend on the magnetic field. And so there will actually be an energy penalty the energy be required to make this transition from this process to that process. And that would again come from the molecular environment, and in this case from nuclear spins that are sitting in the molecule. And so now you would have a process which is driven by fluctuations, from local fluctuations from a magnetic environment, nuclear spins in the molecules. And the resonance frequencies will depend on the external magnetic field. And now you can start to believe that something like this could actually be turned, if all the properties are right, into a magnetic field sensor. And indeed, again, when you do the theory, then you can see when all the conditions are right, this can be a setting which would be sensitive to magnetic fields that are of the strength of the Earth's magnetic field. And it could actually be sensitive to the inclination of this magnetic field relative to the molecules on which this process, relative to the orientation of these molecules that on which this process is happening. Okay, and again, experimentally, we have not proven that this is the case. Theoretically, for a quantum physicist, one would say, yeah, this can happen. No doubt about it. If the conditions are right, this can happen. So the big question is, what would be molecular structures where this can happen? Are all the conditions satisfied that we need for this process to happen? And then can we somehow show experimentally that this is really happening? Okay. And so again, we have this challenge that we have a process that can happen in principle. For a theoretician, it can happen, but does it happen in biology? And so what we really want to do is we want to go also and develop new sensing technologies that use quantum mechanics, actually, to try and be able to examine an individual membrane receptor, and that are so sensitive that we can place this sensor close to one of these receptors and see the motion of a single electron. That's the goal, and that's really quite difficult. But there are new methods that can actually achieve this. And in fact, we have built them so much that in a physics lab environment, they can actually see the magnetic moment of a single nuclear spin, of a single electron spin, and see in principle the motion of these electron spins. What we have not done yet is to do exactly the same thing in a biological environment. That's the next development step. So what I want to show you here is what material that is that we are using, and basically a few examples of what we have achieved here. And so the material that we are using is diamond. And in the end, we will use diamonds that are maybe just a very small diamond nanoparticles that are maybe 10 nanometers in diameter or so. And we don't want to use perfect diamond because perfect diamond is extremely boring. It's completely transparent to visible light. What we want to have is, we want to have actually very, very small defects in the lattice structure, where basically actually the perfect diamond lattice has at one place a carbon atom replaced by a nitrogen atom, and the next lattice site is actually empty. So a carbon atom has been removed. And the effect of that is that there is actually several electrons that are sitting in this space here, and they are localized, they're sitting here, but they have a magnetic moment. And this magnetic moment will react to magnetic fields. And we wish actually then to measure the energy change of this magnetic moment in the presence of a magnetic field, and that makes our sensor. So that's really the basic idea here. We will have an electronic moment here, and we can actually read out its energies with visible light. So we shine a laser onto the system, and depending on whether the electron spin is pointing up or down, so whether the moment is pointing in this direction or that direction, the resonance fluorescence intensity will change. And so we can see the orientation of this one electron spin, and we can read out its energies. And so if nearby there is an electron spin in a biological molecule, maybe 10 nanometers from there, it will create a magnetic field that changes the energy of this electron spin here. And so this is what we can read out. So this is experimentally demonstrated. So this works, yeah. So we can actually take a diamond, we can create one of these defect centers very close to the diamond surface, a few nanometers, 10 nanometers, 5 nanometers from the surface. We can place an individual protein or an individual even nuclear spins on top, and we can see the presence or absence of nuclear spins and of electron spins experimentally, okay? And now we want to use this sensor basically, and the dream, so this is theory again, so we worked out theoretically whether we can take such a sensor, such a small diamond nanoparticle, move it close to a molecule on which we think this magnetoreception, this electron excitation, this radical pair mechanism takes place. That we move the sensor close, that we then make a photoexcitation of this complex, that we see the electrons being generated, and that we see maybe some of the spin dynamics of these electrons. So this would be a very direct proof of this radical pair mechanism that is, that is conjectured to underlie magnetoreception in birds. Okay, and so this is a challenging experiment for sure, but let's say this should be possible given that we have already shown in a physics lab environment that we can even see individual electron or nuclear spins. The same we would like to do with this olfactory receptor. So we would like to go to a cell membrane, either a real cell membrane, or we take such a membrane receptor and encapsulate it in an artificial membrane. We'd like to bring close to it such a diamond nanoparticle, and then try and see how the structure of this membrane receptor might change when an olfactory molecule comes. And we would like to see whether we can actually see an electron transport process in such a receptor, okay? And if we don't see any electron transport processes here, then, well, I'm afraid that this would be bad news for this idea that, electron transport processes might help in olfactory sensing. But if we do see such an electron transport, That would be a strong idea, a strong hint that there might be something to this theory. And then we would have to look more closely whether we can really show that this might depend on vibrational spectra in the environment and so on. Yeah? So this would be an experimental technology that would allow us really to go to a single membrane receptor and see something see some structural changes in it. One may also think of attaching to this receptor maybe some electron spin labels, and then see the motion of the receptor or the change in the structure via the change in the position of these electron spin labels. All right. So these are things that we are really exploring experimentally here, trying to realize. So this is really happening in this building also. And maybe here a small example of what we can do. So this is real experiment now. And so here you see actually a picture of this is a cell. And these red spots inside, this is actually resonance fluorescence in this case, of individual diamond nanoparticles that are inside of this cell. Yeah. So they have diffused into that cell, and we can show really that they're not sitting on top of the cell membrane, they're really inside. One can measure the depth of these particles quite well. And then what we have done is, so these particles are sitting there, and so these are diamond nanoparticles, and they contain one of these electron spins that we want to use for sensing. And here what we have shown initially is that we can really control now this electron spin and its properties inside of these diamond nanoparticles, which sits inside the cell. Okay, and here in the first step, we have not made sensing experiment yet. We have just shown that we can really protect by clever control schemes the electron spin from a lot of the environmental noise that is present in these cells. And now the next step will be that we will make it sensitive to one particular frequency that we wish to see. And we want to, for example, detect in the presence of particular nuclear spin species or maybe electron spins inside of this cell. Okay. And so this is a first step of developing a technology that can, in the end, really make very, very localized measurements on, in the end, individual membrane proteins in a cellular environment. And this will be essential in order to prove or disprove some of these ideas that I've presented here concerning the olfactory sense and the magnetoreception. But it is also a technology that can more generally ask interesting, maybe is able to help biology understand better or examine specific processes inside of a cell at a very high spatial resolution. Okay. And with that, I would really like to close. So this was, I mean, a bit of an overview of, well, questions that we are asking, that quantum mechanics is important, that noise is important, that there are questions that are theoretically, processes that are theoretically possible, but where we don't know yet whether they really happen in biology. And also quantum technologies that may actually allow us to prove or disprove some of these theories. And what I have here is now some references or different levels of complexity, or let's say, so the first one is, I don't know why these both show up. So in the background, you see a European Science Foundation report, and you see a link at the bottom. And that's really a very high level description. We made a report basically a few years back where we sat together and we identified challenges as we see them at present in the field of quantum biology. So what could be interesting areas of application? What do we need in terms of experimental methods? What do we need in terms of maybe theoretical and numerical methods to examine these problems? And this is described at a very high level language, so not too much quantum physics required. And then there is here in the center here in contemporary physics. This is a few years ago. Myself and my wife, in fact, we wrote an article which is, let's say, for physicists, but not necessarily quantum physicists, trying to bring out what are key questions in quantum biology and key challenges there. So these are these two pieces of literature that may be of interest to you. It's the same one again. And at the bottom here, we have, that's a mini review in Angewandte Chemie that I wrote together with experimentalists and chemists. And this is really about these diamond sensors and their potentialities, as we see, for biology. And this was basically the output, or was partly output of an ERC grant that the three of us there had together. And we are very much pursuing this development of these sensors, and of course also interested in seeing applications, potential applications of them in biological systems. And with that, I really want to close. So this is my team. This is only the theorists and some of the funders that have supported this work. So we are very happy to acknowledge funding of the European Research Council. So much of this research was initially funded by a synergy grant, and we got another one, and this means we got 10 million euros or so to spend on, trying to explore these kind of questions. The Humboldt Foundation was extremely generous. They actually brought me from England to Germany again by waving a lot of money in front of me and the opportunity to spend it in any way I like. And I used it to actually push exactly these ideas of the interface of quantum mechanics and biology, because at the time it was essentially impossible to get grants, normal research grants for that, because this was considered too crazy. But the Humboldt Foundation they basically gave me 3.5 million euros and said, do with it whatever you want, as long as it's good science. And so that's what I tried to do. And with that, I would like to close. These are the names and the building. And so now I welcome questions, discussions, whatever you wish. Thank you.
Martin, thank you very much.